Hello, everyone. Today, we're diving into one of the most revolutionary techniques in modern science, PCR, or polymerase chain reaction. Imagine you're a scientist, and you only have one tiny, microscopic piece of DNA to work with. How do you study it? How do we go from that single fragment to millions of identical copies in just a few hours? The answer is PCR. As a cornerstone of molecular biology, PCR makes everything possible, from fast disease diagnosis to solving crimes through forensic investigations. PCR was invented by American biochemist Kary Mullis in 1983, revolutionary for its simplicity. Instead of relying on living cells to replicate DNA, Mullis wondered, why not copy it repeatedly in a test tube? This breakthrough completely transformed molecular biology, earning him the Nobel Prize in Chemistry. PCR is a technique used to amplify a specific DNA sequence. Amplification means making many copies of a DNA fragment. Starting from just a few DNA molecules, PCR can generate millions or even billions of copies within a few hours. To perform PCR, we need template DNA, primers, DNA nucleotides, DNTPs, Taq DNA polymerase, magnesium ions, usually supplied as magnesium chloride, and a thermal cycler. One important question is, why do we use Taq polymerase? During PCR, the reaction mixture is repeatedly heated to very high temperatures, around 94 to 95 degrees Celsius. Most enzymes would denature and lose their function under these conditions. However, Taq polymerase is thermostable. It was originally isolated from Thermus aquaticus, a bacterium that lives in hot springs. Because it can withstand repeated heating cycles, Taq polymerase remains active throughout the PCR process. And magnesium ions, usually supplied as magnesium chloride, are required because most thermostable DNA polymerases need free divalent cations for their activity. Now let's look at the three main steps of a PCR cycle. The first step is denaturation. The reaction is heated to approximately 94 degrees Celsius. At this temperature, the hydrogen bonds holding the two DNA strands together are broken. As a result, the double-stranded DNA separates into two single strands. The second step is annealing. The temperature is lowered to around 50 to 60 degrees Celsius. This allows the primers to bind, or anneal, to their complementary sequences on the template DNA. Primer design is extremely important. Good primers improve both the efficiency and specificity of PCR. Poorly designed primers may bind to the wrong locations, resulting in unwanted amplification or weak PCR products. The third step is extension, also known as elongation. The temperature is increased to approximately 72 degrees Celsius, which is the optimal temperature for Taq polymerase. The enzyme binds to the primers and begins adding nucleotides to synthesize new DNA strands. At the end of one cycle, each DNA molecule has been copied, doubling the amount of DNA present. After extension, the temperature is raised again to 94 degrees Celsius, and the cycle repeats. PCR is typically performed for 25 to 30 cycles. PCR has numerous applications. It is widely used for gene expression analysis and the detection of genetic mutations. PCR is also commonly used to detect pathogens, such as bacteria and viruses, making it an important tool in disease diagnosis. In the food industry, PCR can help identify allergens and detect contamination. In forensic science, PCR plays a key role in DNA profiling and criminal investigations. Despite its many advantages, PCR has some limitations. One limitation is that the DNA sequence surrounding the target region must already be known so that appropriate primers can be designed. Another limitation is that standard PCR can only efficiently amplify DNA fragments up to a certain length. Very large DNA fragments often require specialized PCR techniques. All right, let's see how well you were paying attention. Here is your pop quiz. Question 1. What is the main function of PCR? Is it A, to separate proteins, B, to amplify DNA, or C, to sequence DNA? The correct answer is B, to amplify DNA. PCR is essentially a molecular photocopier that creates millions of copies of a specific DNA sequence. Question 2. During which PCR step do primers bind to the template DNA? A. Denaturation, B. Annealing, or C. Extension? The answer is B. Annealing. During this step, primers attach to complementary DNA sequences and define the region that will be amplified. Final question. Why is Taq polymerase used in PCR? A. It binds DNA more specifically. B. It produces fluorescent signals. Or C. It is thermostable and can withstand high temperatures. The answer is C. Taq polymerase remains active even after repeated heating to around 94 degrees Celsius, making it ideal for PCR. How many did you get correct? Let me know your score in the comments below. If you enjoyed this video and found it helpful, don't forget to like, subscribe, and share it with others interested in molecular biology. Thanks for watching. Stay curious, keep exploring, and we'll see you in the next deep dive into the wonders of science.